Hello and welcome to the Cheshire and Warrington Pledge Partnership on the Learn Live channel. The idea behind the Cheshire and Warrington Pledge is to put employers at the heart of inspiring, informing and communicating with the next generation of employees in the Cheshire and Warrington areas. There are opportunities for a variety of roles such as warehouse operatives, accountants, politicians and computer network architects, and that's just to name a few. These will be featured on the Learn Live channel with the sole purpose to help make you stand out to future employers on the field that you want to be in. Now, so far we've heard from companies such as Apprentify and Franklin Financial, but today we'll be hearing from AstraZeneca's Miriam Guest, Georgia Critchlow and Benjamin Pickard. Miriam, Georgie and Ben will be sharing a summary of working in the pharmaceutical industry and details around the day-to-day working of microbiologists. Our interactions with other scientists and how the principles of infection control rate to manufacturing medicines. Now, if you have any questions, please make sure you ask them in the live chat facility next to the video player. And a member of the team will get back to you and answer any questions that you may have. Also, please make sure you're paying attention throughout the presentation, as this will help you create a winning entry for the competition coming up. Now, over to Miriam, Georgie and Ben.
Thanks, Stuart. So today I have been joined by Miriam, Georgia and Ben from AstraZeneca. So Miriam, if you want to tell us a little bit about yourself.
Hello, I'm Miriam Guest and I'm from AstraZeneca. I'm an associate principal scientist in microbiology and I work in the development function of the organisation. So we're all about bringing new medicines to market, bringing new medicines to patients and turning molecules into those products that we can really help people with.
Hi, I'm Georgia Critchlow. I'm a quality control microbiology apprentice at AstraZeneca. I'm involved in the continuous quality control of the drugs that we produce on site.
That's great, thank you, Georgia. And Ben, if you could do the same for me.
Hi, I'm Ben Pickard. I'm a microbiology apprentice in my third year at AstraZeneca and I work in development with Miriam.
That's brilliant, thank you, Ben. So let's find out more about AstraZeneca.
Okay, so if you listen carefully during the presentation, we're going to have two activities for you to take part in. You get the choice which one you'd like to choose. So when you've done that, you can submit your entries and the team from AstraZeneca, myself, Ben and Georgia, will have a look at those and choose the best from each category. And the best will win a wonderful microbiology themed prize. So activity 1, this is a poster presentation. What I'd like you to do is to research and label the components of a biohazard suit. Annotate the labels to explain the function and the properties of those components. Or you can choose activity 2, and that's to create a flow process chart. That chart will cover the steps that must be followed in order to take a sample swab to the observation of an agar plate result after incubation. So with that, over to the presentation. Thank you. Okay, so I'm here today to talk a bit about what science can do, especially in the area of microbiology. And what we're about at AstraZeneca. As we said before, myself and Ben are part of the development part of the organisation, and George is from our quality control group. So between the three of us, we're generating a lot of data and trying to understand that and apply that to what science can do. So I'm going to start with a bit of an introduction to AstraZeneca to give you an idea of what we're about. Obviously, we've all heard so much about COVID-19 in the news, so I wanted to share a little bit about AstraZeneca's response to that. Linked to the news and COVID, we've seen microbiologists all over the place, and we're going to give you an overview of what microbiology is all about at AstraZeneca, how we work with the different STEM interfaces, and a little bit of an insight into our working lives. So Georgia and Ben will be joining me in this. So at AstraZeneca, we want to be seen leading in science and healthcare to create value and growth. And what does that really mean? That means we want to get medicines to patients. We want to help people and really address some of the challenges that we're living with these days. We're very focused on science. Our sort of tagline is what science can do, and we believe that science should be at the core of everything we do. So in reality, that means we're going out and generating information and data to understand our processes, our medicines, and making sure that they're safe and that they work, that we do the right thing, and that we do everything we can to protect our patients and keep them healthy. Another huge part of AstraZeneca, which is especially important to me, is our commitment to sustainability. We've got an ambitious carbon zero goal to reach that by 2025, and we want to use our capabilities in science to make the most meaningful impact to where society needs it most: health. And I think a lot of people, when we talk about sustainability, we often think about the planet and sort of the green stuff, that environmental protection. But there's another two important pillars of sustainability at AstraZeneca, and that's access to healthcare. You know, we can invent these medicines and create these products, but if we can't reach our patients, then it's for nothing. So we want to make sure we work in a world where we can have access to healthcare. And we invest in those health systems to try and prevent disease as well as cure it. An ethical business culture is really important. You'll see a lot of information on AstraZeneca.com where we try and be open about our business. And of course, environmental protection. As I said, we have the ambitious carbon zero target, but we're doing everything we can to reduce our greenhouse gases, look after our wastewater management, and control our processes as best as we can. In terms of the ethical culture as well, a big piece that's important to me at AstraZeneca is the inclusion and diversity. Having a workforce that's happy to come to work and engage and be active, I think it's important to make sure that we all feel we belong. So letting everyone have a seat at that table, making sure that their voice is heard, really gives us the best opportunity to learn from others. So COVID-19, you'll have seen in the last few days the news that AstraZeneca's working on a vaccine. I won't talk too much about that in these slides, but some of the other things that we're doing in this area, we're looking at our existing medicines to see how they can treat COVID-19. We're researching antibodies to target COVID-19. And in the UK, we've done a lot to support the testing laboratories. We've collaborated with GSK and the University of Cambridge, and we've established big testing capabilities to screen swabs for the virus. We've also had to prioritise our medicine supply chain. You know, we've still got patients that need our medicines, so we're doing all we can to safeguard those supplies to keep our patients' supply of their daily needs met. As I said, access to healthcare is a really important part of sustainability at AstraZeneca. And what we've done in response to COVID, we've donated 9 million face masks to healthcare people around the world. And we have a wonderful programme called Healthy Heart Africa. And this focuses on the control of hypertension and decreasing the burden of cardiovascular disease across Africa. It's a prevention and education programme. But we've reallocated some of the work that we're doing there to help communities in Kenya, Ethiopia, Tanzania and Ghana to help combat the virus with education on preventing, spreading and keeping our healthcare professionals safe there. With that, Ben's going to talk a little bit in more detail about what we're doing in microbiology at AstraZeneca. Ben.
Thanks, Miriam. Microbiology is a relentless battle to protect our products and patients from microbes. Microbes are all around us and they're important to keep us and the planet healthy. But we need to be careful about microbial contamination of our products, as large amounts of microbes could seriously damage our patients who may have compromised immune systems. So to prove that our products are safe, we need to generate data from testing and other reports from things such as manufacturing processes. In development, where myself and Miriam work, we need to show that our drugs are safe for patients to take and also effective in what we want them to do, so that they have the desired therapeutic effect. So We show this through various types of testing in laboratories. We then, in development, our drugs go through clinical trials process, which is split into 3 phases. The first phase being in healthy volunteers, and if we can show that these are safe, the drug then progresses to the next stages of clinical trials with patient populations, a smaller population to start with in phase 2, and then if it progresses to phase 3, a larger patient population. So once we go through the clinical trials process and we generate data to show that the drug's safe to take, and we've got information about the manufacturing processes and the controls we've put in place, we work with different regulatory agencies across the world and engage with them to provide data and meet their requirements, so the drug is of sufficient quality and is safe to go to their markets and be used across the world.
Thanks, Ben. And just for us to achieve that, we need to work across the STEM areas, the science, the technology, the engineering and maths. And we have multiple different sorts of scientists working within AstraZeneca. In microbiology, we're performing evaluations to show that the likelihood of microbial survival in worst case conditions can be minimized and controlled. We have to work with other people. I'm sure at school you're working in big teams to do things, and that teamwork's really valuable in the science area. We adopt new technologies such as augmented realities, so we can understand our manufacturing processes without contaminating them, because as soon as you put a person in that area, you've contaminated your process. So that remoteness that we can achieve by augmented realities is becoming really, really valuable. Our engineering colleagues are fantastic to design processes that will reduce the risk of cross-contamination. And those teams that we work in every day, we all have a different perspective to bring. And one thing that I've never been great at is maths, but I work with our data scientists to translate these huge volumes of data into a way that we can get a clear and consolidated understanding of what that message means and how that translates to our day-to-day activities. So Georgia's going to talk a little bit more about our microbial quality.
So to link the 2 stages together, once the drug has passed all the tests and it's being scaled up and manufactured on a large scale for distribution, there's 3 main areas that have to be monitored throughout its production. So firstly, it's the monitoring of the environment that it's produced in, the water systems that are required for its production, and also the actual drug itself and any input materials that are required. So I'm going to start with the environment. So agar plates, like the ones pictured, are used to monitor the environment in the manufacturing areas. So firstly, they monitor the air. So this can be used using an air sampler machine, which sucks air continuously onto an agar plate for around 45 minutes, and that will trap any microbes. Or there's also settle plates, which will monitor droplets in the air. So any droplets with microorganisms will fall onto the plate and they'll be trapped as well. The second kind is operators, so the people who are physically making the drugs. So this, with each activity that they do, sort of cleaning and anything associated with the product, they've got to take a finger down to ensure that the hands and the gloves that they're using are sterile. And then once a month, they've also got to monitor the gowns that they wear. So these are used with small plates, and they'll take one from the hood, the chest, their arm, and the leg. And then lastly, the surfaces. So firstly, there could be a contact plate, which is a small plate that you roll onto a flat surface, and that'll trap any microorganisms. And then secondly, there's a swab. So a swab it's sort of like a cotton bud, comes with a vial of liquid. So you'll wet your swab, take your sample from it, maybe a curved surface, so it could be somewhere that's hard to reach as well. So you'll take that, and then you'll put the swab onto an agar plate, similar size to the one in the photo, and you'll just swab it on as much as you can onto the plate. And then this plate will get incubated with all the other monitoring at 30 to 35 degrees for 5 days. Then after these 5 days, you'll take out all your samples, open them all, read them underneath a light. So when you're reading the plate, you're looking for any colonies that may have grown. So these could be like small yellow colonies. They're usually circular, which indicates microbial growth. So then all these results are reported into the data systems that we have. So the second kind of microbial poly testing is water. So there's two main types of water that are used in manufacturing. The first is water for injection. So this is being directly used and injected into patients. So therefore, it's got to be sterile. As Ben said, they may have compromised immunity, so any microbial contamination could be very damaging to them. And the second kind is purified water. So this might be used to make up cleaning solutions and things like that. So this has also got to be as clean as possible, but it's not as high a grade water as water for injection. So it can have some microbial limits. So it's got a higher microbial limit, should I say. And lastly, I'll explain actually. So any water samples are taken and filtered through a small filter, and in these filters there's tiny holes, so the water can pass through, but any microbes will be trapped in the filter, and then this filter is rolled again onto an agar plate to make sure there's full contact with the agar, and the bacteria can take up the nutrients from the agar and will grow again into small colonies, and these are read and reported as well. So lastly, the product testing. So input materials and final products have to be microbiologically tested. So there's many different products, therefore many different methods of testing these products, but I suppose one of the most commonly used at Macclesfield is sterility testing. So one of our main products that we produce will be sort of dissolved into saline. This saline will then be filtered through different filters, and then the filters will be incubated in different kinds of broth, and any growth will have to be reported, but generally they should be sterile as they're being injected into patients. So this is sort of the ways that we can control microbial contamination in our manufacturing areas, first of which is PPE. So this includes goggles, hoods, suits, boots, and I'm going to go through the way that the operators suit up. So first they'll go into the area, scrub the hands up using an antimicrobial hand soap. Then they'll do the first layer of gloves. So these are sterile gloves that have been sterilised. And then they'll go on, put on the hood, then mask up and put on the goggles. Then they'll go on to put on the suit. So I think it's bottom first and then the top half. And then lastly, as they step over the next stage, they'll put the boots on, and then they'll do a second pair of gloves and then sterilise their hands using alcohol. So then that's the full gown procedure. So you can see in the image on the left, that's sort of what the gown looks like. And then all of these gowns have to be sterile as well. So they're sterilised off-site, and they'll be sterile when they put them on. So that's why they've got to monitor it once a month to ensure that the operators aren't themselves contaminating the suit while they're putting it on. So this is also for their own safety. So the gloves, with the materials they're using, they could be harmful if you've got them on your skin, things like that. So next is containment. So you may use isolators, which are big machines that you put only your hands into, and the air inside the machine is sterile. So whatever you're doing, it's very unlikely that the operator is going to contaminate it. They also may use laminar airflow cabinets that use filters that filter air and push sterilised air onto whatever you're doing. So this can be used in testing of water and testing of products as well. So lastly, I'm sure you've all heard a lot about this recently, good hand hygiene, using the correct technique for, I think it's around a minute or 2 minutes, is it? I should know that really. And then so you can use alcohol liquid or, as I said, the antimicrobial hand scrub. Then also the use of gloves is important in what we do. So you may need them for cleaning, which you'll need like a lower grade glove that doesn't need to be sterile if it's more protection. Or you may need sterile gloves if you're doing your testing. So this is, me and Ben are going to do a bit of this each. So I'll go through what I do first. So in the morning, you'll go around and collect your samples that may be water, product or environmental. And then you'll prep all your samples and make sure that they're all ready for your testing. Everything should be accounted for. All the details are correct. So you might need to record expiries and batch details of your reagents that you're going to be using. Then you'll perform your testing. In the case of environmental monitoring, you'll go through all your samples and make sure everything's accounted for rather than perform testing, as you're just ensuring that the operators have recorded everything correctly, and you'll record down the details of their finger dabs and the activities they've been carrying out. So that may take you to about lunchtime. And then next you'll have to record the details. So I've gone through that, record the details of your testing. And you might have to second check other people's test details in some cases. So, and then lastly, every day you'll have results to read, like I was saying with the lamp, and reading any colonies that you may have grown. And then these results will be put onto any system and reviewed by a senior lab technician. And then the seniors are responsible for reporting any out of specification results. So you may need like teamwork, which is really important. So you might have done a bit of that in A level in your own. You might be doing that in A level when you go and do experiments in college, and it's really important to get as involved as you can as possible, and just make sure you're comfortable with talking to other people and working with other people. You want to go, Ben?
Yeah, thanks, Georgia. So in development, I work with a host of different drug projects that are in development. So a lot of my day is taken up with meetings. So so some of the communication skills that I learned from a time in education really help with that, and I've learned to develop it since being here for the past few years. And I also get to go in the lab from time to time, which is always nice. Working on different pieces of work for projects, but mainly I'm based in an office, advising on the micro side of things for development.
Thanks, Ben, Georgia. I was just going to share a few highlights from my career. As I said before, I've been doing this for just over 20 years. And I think one of the things that the best thing about my job is when we get regulatory acceptance of a new drug product. We can be working on these compounds for years, 5, 7, 10 years. And that's what it takes to generate the data to show that it's safe and that it works. You'll have these regulatory interactions when you've got this data set, and the regulators will come back to you and ask you questions. And when you get that final letter of acceptance where they say it was considered adequate, it's the best news in the world. It's like scoring a goal in the World Cup final, because a lot of these compounds don't make it that far. We might work on something for 5 years and not get the data we need. So to get those ones that make it, that's a really big high. You're helping patients. Another thing that I've been doing in recent years is going to conferences and presenting and sharing our science at those conferences, and that's fantastic. There's always an opportunity to learn more in AstraZeneca and working in science. So being able to speak and meet other people that are in similar roles is always fascinating. Last year I won a Global Operations Award, and that was quite a nice piece of recognition. I think AZ give out about 6 of these awards a year, so to get one of those was pretty special. I work across the globe, so helping microbiologists from Sydney out in the east to California out in the west is always really nice. And sort of, if ever I'm going on holiday, I've got people that I can reach out to for restaurant recommendations, so that's always good fun. And then getting things published, it's a nice thing to have. So yeah, it's AstraZeneca's, and working in development and working in science, it really gives us the opportunity for that lifelong learning and collaboration. And working with teams and working with other people. And I know that Georgia and Ben have been doing some really exciting things in their career so far. So do you want to share some of your highlights?
Yeah, so I'll go first. So one of my highlights is a project I've been working on for the past year. It's a new technology, so it's been really exciting to sort of fiddle about with that and try it out and see how it works. The technology is to eventually move away from compendial testing that we have in place, the manual methods. So why it's significant to me is the impact that it could potentially have. So it would reduce the number of consumables that we'd use compared to the compendial method. So less single-use plastic, less waste. So there's real benefits for the environment. And from a business aspect, it also reduces hands-on people time and also the data integrity aspect of it. As it's a new technology, it saves it directly onto a system. So both of those aspects make it really a meaningful project that I've been working on, and so I'm proud to say that I've been a part of it.
So I've really enjoyed my third year project where I've been researching into a new method for filtering water samples that I was speaking about before. So at the minute we use sort of like an old-fashioned big, called a manifold. It's a big metal vacuum system. So that uses a lot of energy for autoclave, and it takes a lot of time to autoclave a manifold every week, which is like steam sterilisation. So instead I've been looking at a small vacuum pump, which is a lot faster, a lot quicker to set up, and hopefully should be a lot better. And I've been speaking to global people, we have a micro forum every month, and I got an opportunity to do a talk at that. And then I also got an opportunity to do a talk at a global standardisation of water testing, which is really interesting, getting a chance to speak to different people and just mad thinking that you're speaking to someone who's literally in Australia, China, America, and it's just really interesting, and it's a good opportunity to research into something that's going to be meaningful.
Thank you. And thanks, Georgia and Ben. So thanks for listening. I really hope you enjoyed our presentations today. And just going back to our task that we outlined at the start. So are you going to choose activity 1, which is a poster presentation? Here you will be researching and labelling the components of a biohazard suit. Annotate those labels to explain the function and the properties of those components. Activity 2 is the flow process chart, where we look at steps we need to take from taking a swab to the observation of an agar plate after incubation. I think one thing you might be interested in on that incubation is that we keep our temperature up to around 35 degrees C. So maybe have a think about why we would do that. For the rules on this activities, so if you just use one side of A4 maximum, only include your first name on that piece of work, and then you can complete that piece of work either on your computer or by hand. If you've done it by hand, just scan or photograph it. Email that to your teacher by Monday the 1st of June at 12 noon, and they'll get it over to myself, Ben and Georgia. We'll have a look at those and pick a winner from both categories. Thank you.
Thank you so much, Miriam, Georgia and Ben, for telling us all about AstraZeneca and what STEM opportunities you have available there. It was really interesting to hear all the things that you guys get up to. For anyone out there as well that has any questions, please leave them in the chat facility and the team will get back to you as soon as possible. So thank you, and back to the studio.
Thank you, Miriam, Georgie and Ben from AstraZeneca for joining us today on the Learn Live channel. It was great to hear the information you shared on AstraZeneca's response to COVID-19 and some of the activities required to develop the new medicinal products. We have more pledges coming up soon. On Thursday, 21st of May, we have Richard Hall from Cultural Services, and he'll be talking to us about the great opportunities available within his department. Then on the 2nd of June, we will have Rachel Brooks from Harper Adams University, as she will be telling us who they are and what they're all about. please remember that it's not too late to leave a question on the live chat facility next to the video player. One of our employees who are connected to the pledge will get back to you as soon as possible. And if you're watching this back as a recording, the live chat is always open 24/7. It's safe, moderated and informative for anyone who wants to leave a question to find out how you can become more employable in the future. So thanks for joining us. We'll see you again on the Learn Live channel. Stay safe, everybody.
Welcome to this live chat facility tutorial. We have had a lot of questions recently about the chat facility and how to use it, so let's get into the video. The chat facility is built with students in mind, with a tool that doesn't need to be parent or teacher led. Each of our pages are linked to the person behind the broadcast, even after it's finished, so you can always ask your questions and get a response about the broadcast. So let's have a look at how to use the chat facility. To use the chat facility, the first thing to do is to type your comment or question into the chat box. You can now enter your screen name. Your screen name will show when you type your question or comment. Now you will see the chat facility ask for your email address. As I mentioned before, each of our pages are linked to people from the organisation who can reply to your questions. You can enter your email address, which will not be shared with anyone, to get a direct response from the people behind the broadcast. If you do not want a direct response, then click the button at the bottom. People behind the broadcast can still respond to you if you don't provide your email address. This just means you may have to come back to the page to check the response. You can now speak to the people behind the broadcast and ask any questions you may have, or check out our partner pages to see the companies we work with who you can speak to right now about their current vacancies. You can also check out our careers page, which connects you to employers. Our live chat service is linked 24/7 to employers from a wide range of sectors, all waiting to hear from you. You can leave a question about how to produce a professional CV or how to prepare for interviews, anything work related, and one of our businesses will respond to you directly. You can also access a wide variety of live interactive broadcasts and video on demand with the live chat support on all pages. Now this bit is for the parents, guardians and teachers. The best bit about the live chat facility is that it is completely moderated, so you don't need to worry if your young person leaves a silly comment, as it will only show on their screen and no one else's. Another great thing about the chat facility is that it is GDPR protected, so no information is shared with anyone else. We will protect all email addresses and will never share personal information of the young people. If you have any questions about the chat facility or the Learn Live channel, please use the email us icon at the top of the page. Thank you, stay safe and stay tuned.